has been something I've been looking forward to. So with that, I'll begin. My study is really kind of morphed into looking at how plant trait variation affects arthropod community ecology. And as we start out, I want to give an intro to plant defense against herbivory. Herbivory is the most common interaction among multicellular organisms terrestrially, and it's very fundamental in shaping entire ecosystems and also in plant-insect interactions and the consequences those have. Plants have evolved numerous ways to defend themselves, and we have understood that over many decades of research. Some of those ways we can categorize them in a bunch of different fashions, but I'll categorize them in this way. We have direct defenses, and direct defenses are defenses that would disrupt the behavior or physiology of the herbivore directly. And so this could be through chemical compounds in the leaves. It could be through structural features such as thorns or trichomes on the leaf, anything that makes it more difficult in order for that herbivore to consume that plant material. And then there's indirect defenses, which also play a role in defending plants against herbivores, except in this manner, what plants do is they would recruit predators of the herbivores, whether they be predatory insects, parasitic insects such as wasps through the release of volatile organic compounds from the leaves that might attract these wasps, or through means of extrafloral nectaries, which might attract ants to particular plants. And then once the ants are there, they would then defend the plants against herbivores. And so these are kind of the two main avenues of research in the plant defense world. That really kind of shaped a lot of the questions that we sought to address. When it comes to plant-herbivore interactions, three avenues of many that are pursued is in regards to ecosystem functioning. And so in this first panel on the left, you can see kind of a stereotypical food web of sorts. And so if you look at how plants and herbivores might interact with each other, that has consequences not only for those plants and the herbivores, but also for maybe entire communities. The predators that feed on the herbivores that might compete with other herbivores for access to resources, et cetera. So that's one is with regards to ecosystem functioning. That's one way that we might try to understand the importance of these plant-herbivore interactions. Another is through evolution or adaptation. In evolution and adaptation, we might look at plant defense compounds and ask questions about how these plant defense compounds may have evolved over time to provide an adaptive mechanism for these plants to then deter herbivory or limit it either directly or indirectly. And then another area of research would be through agriculture. In agriculture, we might have many different organisms of a particular plant species, an apple tree, let's say, and we might be selecting for certain individuals that might show greater resistance to herbivory just naturally, and then propagate those as a means to increased harvest yields. And so these are kind of these three areas of many in plant-herbivore interaction research. Now, when it comes to traditional approaches used to try to address questions in plant-herbivore interaction, this kind of tries to summarize, I guess, the main one that's used. We look at herbivory creating some selective pressure on the plants. Herbivores and plants are antagonistic. And so as plants are consumed, their fitness level drops. And so we might think, oh, that's a selective pressure for these plants then to adapt to deter herbivory, to resist it through some means, maybe directly or indirectly. So adaptation through evolution of defense. And then we assume that adaptation can be measured, or it manifests itself in individual plant traits. And so we might measure aspects of a plant and say, hey, these plant traits might give us insight into how a plant has adapted to prevent herbivory or deter herbivory, to resist it. And so that's a traditional approach to looking at plant defense research. And while it's very beneficial, and I think it has produced quite a lot, there's, I think, obviously some critiques, as with any approach to research. And here's two critiques, and then kind of a summary of that. Often the focus in plant defense research is on these highly specialized interactions. And so we might be interested in this one particular insect herbivore species and this one particular plant. And while that may be true for that particular system, and that may be relevant in that context, we think that many or most interactions between plants and herbivores are not that specialized. Many herbivores are generalists, whether they can consume plant material from species within a family or across multiple families. And so focusing on highly specialized interactions may lose some of that context in what we might think is going on in natural settings. And another critique is that a lot of these studies will often use controlled or simplified conditions in order to try to get at or address these questions. And so in this bottom picture, you have a plant and an herbivore in a greenhouse setting. And so when you put things in a greenhouse, you are controlling, by and large, the abiotic environment. Not entirely, but a lot. A lot of the abiotic environment is controlled for. And you might be controlling soil conditions and water availability and all these different aspects that are not natural. And so you might be able to deduce conclusions from this research, but I think that this kind of research really lacks relevance to realistic ecological settings. So there's constraints to it. And what we sought to do is to look at these constraints and try to design a project and research questions that allowed us to highlight these constraints and look at this question without these constraints in place. So our guiding questions really that shaped our research are two of them. Is there evidence that plants are shaping their associated communities, in particular herbivores, but also in general the entire arthropod communities? So what evidence exists that plants are actually shaping these communities? And then the second question is, if so, are the traits that drive this evolved for that purpose? In other words, are the traits that are driving the shaping of these arthropod communities plant defense traits? Are they evolved for plants to defend themselves against herbivory, or are other traits involved? Other traits that we might look at as non-defense traits, which we'll get to in chapter 3. So a little bit on the study system. Here is an overhead picture that you can see of the common garden that we constructed over at Concordia University in Irvine. We have 20 plots. In each plot, we have 14 different plant species. These plant species are native to coastal sage scrub ecosystem, and they all have very similar growth forms. And we thought that the advantage of doing it this way is that we can look at whole plant communities. So we're looking at a macroevolutionary approach. This approach is comparative, so we can compare between multiple plant species and then compare between years, right? It's realistic, it's outdoors, so we're not controlling really anything here. And it took place over 3 years. And so we have interannual variation that is folded into this, and it's a single ecosystem. So we're not looking at multiple ecosystems along, let's say, a latitudinal gradient or from shoreline inland. We're looking at everything in the same space, same time. And so we feel like this approach will help us really address some of maybe some of the blind spots of traditional ways of approaching these questions. All right, so here's the roadmap of the different questions that I'm going to talk about today. Chapter 1, the question that I really focused on is, to what degree do plant traits that affect resistance to herbivory drive herbivore density? And so here I'm really focused on these direct defense traits in plants and how they kind of sum together to maybe influence or drive herbivory in our system. And then in chapter 2, what effects do these plants have? Do plants have on predator mediation of herbivory? And so here I'm looking at the indirect effects from that figure that I showed earlier. And then the last one is saying, okay, well, what other traits might be involved here? Non-traditional traits studied in plant defense research. So do plant traits adapted for response to the hydrologic environment? So water traits. Do they affect herbivory, and if so, how? Okay, so those are my three questions. And so question number one, the methods that we used to try to address this question was really looking at a single assay that is very commonly used in defense research, and it's called a generalist bioassay. It's generalist because we're using a generalist herbivore that can consume many different species of plants, Spodoptera exigua, and we will take that herbivore and we'll put it in a laboratory setting, and we'll feed that herbivore leaves of all the plants in our study system. And then we will measure weight gain, mass change over time, and assume that herbivores or the Spodoptera that gained the most mass did so because the plant was least resistant to herbivory. And so we're making that assumption there. And then what we did is we simultaneously collected herbivore density in the field. And so we literally took a vacuum cleaner and vacuumed all the plants every year. And we classified all of the arthropods from that and just looked at the herbivores for this particular question and asked, what is the herbivore density on each plant for each year? And then after that, we compared the two. And we looked at the correlations between herbivore density in the field and mass gain Spodoptera in the lab, with the assumption or the prediction that if plants have high resistance to herbivores, if they have high amounts of direct defense, right, strong direct defense, then we should have low mass gain on the Spodoptera, and we should simultaneously have low herbivore densities in the field, right? And so here's the results. First, we needed to determine whether or not there's variance that exist within mass changes in Spodoptera, as well as herbivore densities across all of our species. And so for this particular panel on the top, these species are denoted by initials there, and we are looking at 2021 levels or mass changes, 2022, 2023, and then all years combined. And so just by looking at this, you can see that there does seem to be variation that exists between mass change in Spodoptera, both interannually, as well as according to species. And so the next panel down, that's looking at herbivore densities across these plants for that same time period. And so herbivore densities do change as well. And so we showed that there is significant variation in Spodoptera mass change, as well as herbivore densities in these plants. And we assume then that that variation is because, or at least some portion of that, is driven by plant resistance. So plants might be resisting these herbivores in different manners, okay? And so what we did then is said, okay, well, what is the correlation that exists between Spodoptera and natural herbivore density in the field? And if we look here, we were a bit surprised that there was very little evidence to suggest that that is actually true. So the big takeaway from here is that while variance in Spodoptera exigua mass is predictive of variance in herbivore density, it is so only for 5 species of our 14. And if you look over here at these R² values in the green highlighted areas, it's quite low for most of them. So the variance in herbivore density, as explained by variance in Spodoptera mass change, is pretty weak, right? So there wasn't a whole lot of evidence for this. And so we then asked the question, okay, forget the individual species, let's lump everything together and look at this from an entire community perspective, right? And if we look at this interannually, how does the changes between average Spodoptera mass and average herbivore densities across all plants, across all species, change each year, okay? And the way to interpret this graph is to look at this red line, which is in 2021, and we see that the Spodoptera mass and the herbivore density are both at the highest, all right? And then as you go through from 2022 all the way to 2023, you see that the Spodoptera mass does seem to decrease pretty significantly over time. And so we interpret that as meaning that these plants are resisting Spodoptera as they age. These are the same plants just over years. And that maybe that there might be some convergence here as more time goes on, possibly, where plants will become more and more resistant to Spodoptera, and that resistance to Spodoptera may actually map onto herbivore densities to a greater degree. But at least right now, we don't really see much of that. And so our takeaways for chapter 1 is that direct resistance does not appear to drive herbivore density for most species in realistic ecological settings for our time frame. And so then we ask questions like, what other factors might be involved in this? And so we then we're interested in, all right, well, we're going to look at indirect defense. And so we think maybe indirect defense is what's primarily driving this, or we might show evidence for that. So we then move on to chapter 2. In chapter 2, then we're interested in what is the effect plants have on predators' mediation of herbivory. And so looking again at indirect defense. To do this, we followed a similar pattern. We quantified herbivore and residual predator density in the field, so vacuumed the plants, and then this time we added predators to the mix. And then in order to get or address bird attacks or predation from birds, we used a clay caterpillar model that I'll describe in the next slide to quantify bird attacks. And then after we did this, we then examined correlations between herbivore densities and predatory arthropod densities, as well as herbivore and bird attacks. All right, so a couple other things that I think need to be mentioned is that we use residual predator arthropod densities in our calculation because there was a very strong relationship between predator and herbivore populations. And so the residual just allows us to say, like, how much more predators exist on any given plant than their relationship between predators and arthropods would predict. And so those residual densities are important because then we use those terms in order to measure that indirect defense from predatory arthropods. For the clay caterpillars, what we did is we just did this with clay and made little caterpillars, me and many students, and we glued them to branches of all of our plants. And we went out for 4 weeks each week and assessed predation by looking at little markings that were present on the caterpillar. So in this particular caterpillar at the top right, you can see this triangular beak-shaped marking. And so then we assumed that that was a bird attacking that caterpillar, and that's how we measured attack rates there. All right, so what are our results? Again, we did want to test for variance. All right, we wanted to make sure that the variance existed in predator densities as well as bird attack rates, because if variance does exist, then we assume that at least plants are selecting for some of that variation. So species do vary in indirect defense from predatory arthropods. There's very strong evidence for that. But interestingly, there's no variance in bird attacks. So plants do not differ in terms of bird attack rates, which may suggest that plants are not recruiting birds indirectly for defensive purposes. Birds may be attracted to other aspects of the plant, but not necessarily for bird attack against predators. So that was our first thing that we looked at. And then we examined correlations between herbivore densities and predatory arthropod densities, as well as herbivore densities and bird attack rates. And we found nothing. No correlation exists between the two. And so based upon this research, we conclude that plants do vary in indirect defense from predatory arthropods, but that variation does not appear to affect variation in herbivory. And another conclusion we make is indirect defense from birds is consistent across species. And so birds really don't seem to be attracted to plants for defensive purposes, or at least plants are not necessarily attracting birds for defensive purposes, is the conclusion we would draw from that. And then finally, indirect defense may be at work, but the effect seems to be minimal, at least in our ecological setting for our time frame. All right. And so after that, then we was like, all right, kind of scratch our heads and went, well, what other traits might be involved here? Do plant traits that are adapted for other purposes, responses to the hydrologic environment in our case, do they affect herbivory, and if so, how? And the reason why we chose plant water traits is because these plants exist in an arid environment, and water as a resource is fluctuates. It's in flux quite a bit, both within a year and then interannually. And so we chose those traits and did some research on them and tried to assess if there's any relationship between those traits and herbivory and total arthropod community as well. So again, back to the vacuum. So we keep vacuuming these plants over and over. And in this time, we're collecting all the insects here and classifying every one of them. And so over 3 years, we had about 55,000 insects that we classified down to taxonomic order. So it's quite a large sample size. Thank you to everybody in this room who that's so many hours. And then the next thing we did is we measured different hydrologic traits of these plants. And see, these are traits that are typically looked at as adapted for the plant to its hydrologic environment. So they're plant water traits. So involved with maybe hydraulic conductivity or photosynthesis or water use, and not necessarily ascribed to their defense purposes, right? And then we looked at correlations between variation in these traits, plant traits, and the composition of the arthropod communities. And so, again, we first looked to determine whether there's variance that exists within these traits among plant species for these plant water traits. And the answer is yes. And there is strong variance, a whole lot of variance that exists in these hydrologic traits. So they vary in hydrologic traits and arthropod community composition, right? So there's a lot of variance that exists between these species. And this then again to us suggests that the plants are adapting to their environment, and they're adapting through these traits in particular. The next thing we did then is to take the variance in all of these plant traits in multivariate space and the variance in the total arthropod diversity, community diversity, and we created what's called ordination plots, right? And so ordination condenses multivariate trait data into a couple dimensions. In our case, we did it into 2 dimensions. And what is seen here is these dots that are colored in. Those are the centroids for the trait variance of the arthropod community variance for each different species. And so you can see again, a lot of variance that exists here. And on top of this, layered on top of this are influential traits and vectors that correspond to the direction of influence that these traits have in the variance on this community. So the centroids correspond to species variance in arthropod communities, and the vectors in panel A represent the relative influences of these plant traits on total arthropod variance. And then in addition to that, we looked at in panel B, vectors that represent the relative influence of individual arthropod orders on total arthropod variance as well. And so there's a lot going on here, and I'm going to take this step by step and try to explain some of the takeaways from this ordination plot. So the first question that we can ask here is, how do traits, individual traits, shape arthropod communities? And so we might take an example of a trait, relative growth rate here, and ask the question is, how does relative growth rate affect the total variation in this particular arthropod community? And the data for that is pretty strong that it does. Most hydraulic traits individually and all hydraulic traits or hydrologic traits combined influence arthropod community composition. So we see that percent water content, for example, is significant, but this R squared of 0.4 just means that 4% of the variance in arthropod community composition is attributed to percent water content as a trait value. And then we can go on down the line. Overall, these traits contribute 13% to the total variance in arthropod communities. And so there's strong evidence that these traits matter. And then the second question that we can ask is, how do these traits combine, sum all the trait variance together? How do they shape individual arthropod orders, right? And the answer to that is very strong. And what we have here in this figure is first looking at the different arthropod orders that are strongly influenced by plant traits. And so these names may mean nothing to you. In parentheses, I put H, O, or P. H is herbivore, O is omnivore, and P is predator. So just to look at their feeding guilds. And so Sternorhyncha and Formicidae, those would be ants and aphids. And so we see here in this top quadrant that ants and aphids are both strongly influenced by plant trait variants and hydrologic traits. And they seem to map onto increased percent carbon concentrations in the leaves. And so as percent carbon concentrations maybe go up in any particular plant, or if you measure a plant with high percent carbon concentrations, we might then predict that it may also have high aphid and ant trait variant in that sample, right? And then we can kind of go on down the line. And what we see then is very strong associations all the way down between plant traits and these different arthropod orders. And what we've done then is we've broken up these arthropod orders and plant traits into different quadrants. And so we can maybe say here that the percent nitrogen, percent water content seem to map onto the Echinirhyncha and so forth. So it's quite interesting and unexpected for us to see these results. And so, you know, after kind of scratching our head for a while and thinking about it and talking about it back and forth, you know, we came up with a conclusion that yes, very strongly do these hydrologic traits seem to cause variation in both herbivore concentrations in plants as well as the entire arthropod community. So they're shaping this arthropod community in pretty fundamental ways, and they're doing it in much greater magnitude than traditionally studied defense traits, both direct and indirect defense traits. And so then we kind of come to our overall conclusion. So what does all this mean for us? Well, we conclude that plant direct and indirect defense does not explain much of the variation in herbivory. While hydrologic traits do show strong effects, not only on herbivory, but the entire arthropod community composition. And the big takeaway for us is this here. We think that this suggests that the effects of resistance, of plant resistance on herbivore abundance, we believe that that's real, that exists. Plants do have defensive traits and they are resisting herbivores. However, this effect may be occluded by the effects of ecological forces shaping herbivore populations through these non-defense traits for small timescales. And this is a graph that attempts to demonstrate just that, is that, you know, maybe we looked simply at these first 3 points here on a very, very small time scale. And so we may not see much effect of resistance on those small time scales as these plants are very young when they were planted. But maybe as time goes on, we may see greater resistance in these plants, and those resistance traits may actually appear and become less occluded by the interannual variation from these ecological forces in shaping these arthropod communities. So with that, I'm going to conclude my talk by giving a couple thank yous to the people in the room. So the first is I want to thank my family, Tracy and Eli and Zoe. You guys have been really patient with me, like way more patient than I deserve. And through this whole thing, you encouraged me to do it. You supported me in it. And you gave me a lot of love and support. And so I'm really grateful for that. And you know that, but it does need to be said. And then to the administration at Concordia University, they provided financial and institutional support for me to participate in this project. I'm a very atypical grad student, as Kaylin mentioned, and so I really appreciate that. I couldn't have done it without their support. To my department, you guys have been so flexible with me and my schedules, because I had to take classes here, and you rearranged my schedule so that I could do that here. And you gave me so much good feedback and challenged me in ways that maybe I didn't want to be challenged, but needed. You were just unending in your care for me and the assistance that you provided with the project. And I'm so grateful for that. To all the field assistants in the room online and those who weren't able to make it, I counted up, there's over 150 of you. who helped out with the project over 5 years. And I would estimate probably about maybe a couple thousand hours at least was spent on this. Thank you for the time, the patience that you gave me as a mentor, and the efforts that you provided, because this would not have been possible without you. And I really am indebted to you big time. And to Sarah and Travis, thank you so much for being on my committee. And you shaped me into a better ecologist. I think I'm a better ecologist, but you shaped me nonetheless. And for that, I'm just so grateful for your questions, for helping me think differently and challenge me when something I said didn't make sense. And I'm so appreciative of that. And then lastly, I just want to thank you, Kaylin, because outside of my close friends and family, there's 4 people who give me a chance. Either because when I didn't deserve that chance, or when it came with great risk. And you're one of those 4. Patricia Scott, Donald Michaels, Rod Soper, and Kaylin Mooney. And I'm so grateful. I'm so grateful that you put me at the center, that you just were so patient with me in so many ways. And you guided me phenomenally. You put me at the center of everything. And I couldn't have done this, I think, with any other advisor. You were the advisor for me, like the only one in the world. And for that, like I owe a debt of gratitude. And so with that, I will conclude my presentation and look forward to taking any questions that you might have.
Let you field your question.
Okay.
And I'll interject if there's some coming on Zoom.
Perfect. Yes.
So you said that these plants within like herbivores, they recruit insects which are natural enemies of the natural enemies of the insects who are attacking the plants.
Correct.
And so how do the recruited insects really benefit? What do they benefit?
So the recruited insects would benefit just through a food source as a herbivore to consume. So if it's a parasitic wasp, this parasitic wasp would then lay its eggs on the herbivore, and the eggs would then hatch and consume the herbivore and use that as a food source. And so that's the main way, that they might benefit. But with the extrafluorocnectaries, the ants would benefit because the plants, we think, are producing a sugary substance and secreting that for the ants as their food source. And in turn, the ants might defend the plant from that. There's also examples, a famous one of ant and acacia domatia. And so these domatia can serve then as housing, as little ant condominiums that the plant might produce. And so then they provide a structure for this ant to live and protect it then from its enemies. And then the ant in turn would protect the acacia tree from herbivory. Yes. Yeah, and me too. Wasn't sure there for a moment. Dicey.
Wondering if you had a chance to think about how your results are back to chapter 11. You see all this variation in the herbivory on the same plant, but year to year.
Yeah.
So do you think that, I imagine you didn't measure these traits every year, but do you think there's variability with the traits you measured in chapter 3 over those years, such that might explain some of it?
Yeah, the traits that I did measure annually were SLA, specific leaf area, and relative growth rate. But there's just 2 measurements because year to year. But I didn't look at percent nitrogen or carbon, just because that's cost prohibitive. So we just only did that once. And so SLA did vary year to year. I didn't really look to see then how SLA mapped onto herbivory in 2021 versus how it mapped on in 2023. But I think that, yeah, that would be a great extension of that.
How much did rainfall vary over those 3 years.
It did, yeah, it did quite a bit. And then there was another aspect that I didn't mention of the study is that I'd originally done this as a precipitation manipulation experiment. And so I covered the entire plot with weed barrier and found out a year and a month into the project that the weed barrier was preventing soil evaporation. And so I'd normalized water moisture levels across the entire plot while thinking I was manipulating them. So, so that definitely influenced things, but you know, at least it was consistently applied to everything. And so, but yeah, there was strong interannual variation, and not just like total precipitation amounts, but also monthly differences. And so some days, like some years we had really wet Decembers and other years we had very dry Decembers. And so even that, I think, has strong potential to influence the plants. It's how did it? I don't know. Because I only took annual measurements at peak growth in May. And so I'm just summing up the entire year. So yeah, it is interesting. And then the ontogeny of the plant too is like, you know, plants that they're 1 year old when they were put in the ground. And so they're not 1 year old anymore. And so they've really developed a lot of woody stems and all these different physiological changes that I think do influence things. And so we're continuing the project and hopefully, you know, as time goes on, maybe the story becomes a little bit clearer, or maybe not, it just becomes even more messy. So that's exciting too. Yeah. Great. Yes.
A lot of these words are foreign to me. First, thank you for that. Very good job. I'm trying to understand the relationship between the herbivores and the non-herbivore arthropods. So, first of all, were all the non-herbivore arthropods predators?
No, so we just looked in chapter 2, we looked just simply at predatory arthropods. And we, you know, with the assumption that if the predatory arthropods are on the plant, it's because the herbivores are there for them as a food source. And so, you know, that's obviously a big assumption to make because maybe that's not entirely true, but yeah.
Yes, I wonder if looking at the correlation between the herbivore populations and the non-herbivore populations will help.
Yeah.
I mean, almost like your conclusion in chapter 3 is that, you know, non-defense related mechanisms are at play here.
Yeah.
And so comparing those two, because if it's non-defense related and you have arthropods that are not either herbivores or, you know, consumers of herbivores, then that might be a way to get at that question as well, or at least further flesh out.
Yeah. Good thoughts. Thank you, Mary. You want to take a question online and
There's lots of congratulations.
a lot of clap hand emojis? No, it's okay. Any other questions? Yes, back.
And the adversities that you encountered along the way, how does that shape your perspective of research?
I came in and I think Kaylin and Travis and Sarah can attest to this is, you know, I came in with these kind of very formulaic, very black and white kind of views on ecology. I think a lot of that is shaped just how I was educated in it. It's, you know, this is interspecific competition. This is not, and so there's these clear lines, these delineators, and now it's all gray. It's like just like it's all fuzzy. And so I think that like being okay in that space and feeling comfortable in the gray, I think is one of the biggest changes in my life, and my department too can attest to this because they've challenged me in that way as well. And I think that would I think would be the biggest takeaway for me to address that particular question, you know, and how it shaped me and just being comfortable in uncertainty. And yeah, that was really new coming from, you know, just a background in sciences. So yes.
Generalizable just other arid climates, or would you say it's just for the specific community in Concordia? And how would other researchers benefit from this?
Great question. I think it's pretty generalizable to coastal sage scrub ecosystems. We did use 14 species native to this area that do show strong phylogenetic differences among them. So I think that the applicability for this particular ecosystem is there. As to whether or not that applies to other arid adapted climates, I don't think, I'm not as certain of that. And so I would be very, very careful to not assume that like this has strong applicability outside of this system unless I came across some evidence that would suggest otherwise. But I think what this allows us to look at is say, okay, well, what are these plant traits that we may not think are involved in defense in these other ecosystems, these other climates, and try to test them and to look at the associations between those plant traits and those non-defense traits in those ecosystems, and look at the associations with the herbivore communities and to kind of see what comes out. And it may be true that in these in ecosystems where it's particularly stressful, you know, if we define stress as, let's say, flux in a resource or lack of a resource, then you may find that these traits matter more than maybe in a system that might be pretty constant in terms of the resource availability, let's say. But I'm just hypothesizing that. Yes.
Really cool project. 150 students is a ton, but it's like a lot of impact. Did you worry about the standardized, did you have a hard time standardizing the data collection?
Did I worry about it? All the time. There's like my signal to noise ratio, I think it's like nothing that anybody's ever seen. But what I tried to do, what I was very careful to do, is try to standardize the methods and be present as a mentor and at least teach every student once on what those methods are and be in as close a proximity to the students as possible as I was going through. And then the other thing I did is try to identify particular students who were showed very strong leadership skills and interest. And so if they're interested, invested, and they seem capable of leading groups, then I would put them kind of in charge of subgroups and divide it out that way. But it definitely was a task. I mean, but a fun one. Like I designed the study for that purpose. I wanted to influence as many undergrads with research opportunities as I could. And that really happened both at UCI, and I was able to mentor several students in excellence in research projects, as well as other opportunities, and Concordia. And that was a treat. And I had a bunch of colleagues come along for the ride too. So, thank you. Yes.
To the slide just after the ordination plot where you had all of the different orders of insects.
Yes. This one? Yep.
What's the importance in percentage of carbon with the plants in relationship to Sterner Indians?
I don't know, but that's like the short answer. But the way that we would typically think about percent carbon in leaves is if you have a lot of carbon that's present in a leaf, that might mean that plant is investing that carbon for longevity purposes. And so it may not be growing really fast. Nitrogen is typically looked at as indicative of high photosynthetic rates, whereas carbon is looked at as saying, hey, maybe cool down on the photosynthesis and let's keep this leaf around a lot longer. And so maybe you find that in plants that are more arid adapted or slower growing, that's typically how the story goes. As to what that has anything to do with Sternorenca, I don't know. I really don't. But I think it does bring up the question of like maybe there is some relationship here. And maybe relationship is not so much the traits, but maybe just looking at this ecological forces causing flux from year to year to year, and how those ecological forces may be shaping the community in ways that these plant defense traits are not, at least for that time scale. So, yeah.
I think I'm going to let Tyler off the hook, although
Okay.
he has a few minutes to for you to congratulate him in person if you'd like, and then there's the reception outside, which Tyler has a couple minutes to go to, but then at 2 o'clock we'll meet him back. So let's congratulate Tyler on the and come up and say hi to Tyler, and then go get some food, and then say hi again.
Thank you, everybody, for tuning in online. Appreciate it.